My heartiest welcome to our next speaker, Michael Levin. He's from Tufts University. Michael Levin is the Vannevar Bush Distinguished Professor of Biology at Tufts University, an associate faculty at Harvard's Wyss Institute, and director of Allen Discovery Center at Tufts. He has published over 400 peer reviewed publications across developmental biology, computer science, and philosophy of mind. Wonderful. His group works to understand information processing and problem solving across scale in a range of naturally evolved, synthetically engineered, and hybrid living systems. Dr. Levin's work spans from fundamental conceptual framework to application in both defects, regeneration, and cancer. I welcome to Professor Michael Levin for his talk on bioelectricity as an interface to conventional intelligence from philosophy to biomedicines. Please, Levin, sir, you can start your presentation now.
Thank you very much. Let's see, I will share my screen. All right, can everybody see the slides?
Yes.
Perfect. Okay. Yeah, and apologies. I think I've changed out the title a little bit as I played with the content of the talk over the last few days. Okay, if anybody's interested in following up on the things that I'm going to talk about, you can find all the primary data, the software, everything else at this webpage. And then my own kind of personal thoughts about what all of those things mean are on this blog here. So I'd like to start with kind of a view that is pervasive in at least the modern Western science conception of what we're doing here, which is shown by this ancient piece of art called Adam is naming the animals in the Garden of Eden. And so what we need to do, I think it's very important to move beyond a few limitations that are expressed in this view, this idea that there are very firm, discrete natural kinds that represent the different sorts of possible animals. Adam is, of course, separate from these, and he knows exactly what the different animals are. And I think we have to move beyond that. And as I'm going to argue, simply adding evolution to this story does not go far enough in what we need to do. But there is something profoundly important about this way of thinking, which is that in the original story, it was Adam's job to name the animals. Okay, God couldn't do it. The angels couldn't do it. It had to be Adam to name these animals. Oops. Sorry, hang on one second. Yeah, here we go. And the reason that this is important is because in these ancient traditions, naming something is actually being able to discover its true nature. Giving something a name means you've understood the true nature of this. And as I'm going to argue, part of our job here is to understand the true nature of a variety of novel beings that have never been here before. And I think this is very important going forward. Okay, the place to start is simply to realize that whatever we say about humans, and there's a lot of philosophy of mind that is written about humans, and what they have in mind is an adult, a neurotypical adult modern human. And we need to understand that whatever we say about this human, it actually stands in the middle of a very smooth continuum. In fact, two continua, both on the developmental scale and the evolutionary scale. Where smoothly and gradually we got here by scaling up the competencies of individual cells. And in fact, I'm going to, towards the end of this talk, I'm going to enlarge this quite a bit by adding another axis to this. But whatever it is that is true of us, we need to understand how it got there, starting with this a much more humble beginning of single cells. So what I do in my work is try to develop a way of thinking about very diverse intelligences. My framework is attempting to help us recognize, create, and relate to a really very wide variety of minds, and this includes familiar creatures like birds and apes and so on, maybe octopuses or whales or whatnot, but also a lot of very weird organisms such as colonial types of systems, swarms, and so on. Of course, engineered new life forms, so new synthetic biology, artificial intelligences, whether software or robotic, and possibly someday even exobiological agents, because the Earth and everything on it is really just an N of one example of what life and mind can be like. The goal of my framework is to allow us to think about all these things at the same time, and not try to draw artificial distinctions between them. And it's very important that this framework move experimental work forward. We are not just doing philosophy. We insist that the formalisms that we develop and the techniques that we develop are designed to move research forward. That is, to develop new capabilities in biomedicine and in robotics and bioengineering and so on, but also very importantly to derive a better ethical framework for enabling us to relate to all these beings. And I'll talk more about this at the end. And so, of course, I'm not the first person to try for something like this. So here's Rosenbluth, Wiener, and Bigelow back in 1943 trying to show the scale of how passive matter can become progressively more functional and more complex to give rise to second-order cognition, such as humans enjoy. And my framework, at least the first part, the 1.0 version of it, is described in this paper if you want to see the details. So I'm going to talk today in three parts. First, I want to show you some unusual biology, which kind of is designed to shake us out of the standard biological systems that the people work on, because those tend to facilitate a very specific kind of thinking that I think we need to break out of. Then I'm going to show you a specific example of applying these kind of ideas using the bioelectrical interface to manipulate and to relate to the collective intelligence of cells in the body. And then at the end, I'm going to say some very speculative things about where the patterns of body and mind actually come from. So let's look at some unusual biology. The first thing that we can talk about is this planarian flatworm. So these little flatworms, they have a brain, they have a central nervous system, a lot of the same neurotransmitters that you and I have, and they have this amazing capacity for regeneration. So you can cut this animal into pieces. The record is something like 275 pieces, and each of these pieces will regrow a perfect little worm. Okay, so there's this kind of almost kind of holographic property where every piece knows exactly what the collective should look like and is able to rebuild it. But one of the other remarkable things about this is because these animals are smart, they can learn. You can do memory and regeneration in the same model system. That's quite rare. And if you do that, so you have these worms, you train them to look for food in these little bumpy discs here. This is place conditioning, so they remember that this is where they get fed. And then you can amputate their head, including their brain. So this tail sits there doing not much for about 8 days until it grows back a new brain. So the brain is essential for behavior. The tail by itself doesn't do anything. Eventually, the brain grows back. And then what you find is that they've recovered the original information. Okay, which tells you several things. It says, first of all, that the actual learned information is not entirely in the brain. It's also somewhere else, perhaps in the rest of the body, for example. Another thing it tells you is that during the process of regeneration, that information is imprinted onto the new brain tissue. So as the new fresh brain comes up, the information is imprinted on there. That may have implications for human patients with degenerative brain disease in the future when we put, let's say, stem cell therapeutics to restore parts of the brain. What's going to happen to that patient's personality, the decades of memories? It looks like it at least in this species, it can be re-imprinted onto this new brain tissue. And so what this is telling you is that information can actually move through the tissue. Now, one of the things that you'll notice here is that there's a very clear and very strong memory of what the pattern should be. That's what allows these individual pieces to regulate back to a correct worm. And in fact, these animals are not only cancer resistant and regenerative, but they are also immortal. They don't age. They don't have a lifespan limit. They just continuously regenerate themselves. So you can see that knowing the anatomical structure of your body is extremely useful. But where do these anatomical structures actually come from? How are they defined? Well, development is incredibly reliable, and you might get the feeling that, well, it comes from the genome, and we know exactly what the genome does. After all, every year you see billions and billions of these acorns give rise to oak trees, exactly the same leaf pattern. And development is so reliable that you might get the feeling that, okay, this is the kind of thing this genome knows how to build, and that's exactly what it builds. And you can have this kind of a hardwired idea about how development proceeds. But along comes a bioengineer, and it happens to be a non-human bioengineer. It's this little parasite, this little wasp, and the wasp teaches us something very important about the material. It teaches us that what the material does under normal circumstances, that is the reliable default development, isn't the whole story of what the material is capable of. And so what the wasp is doing is it prompts the plant cell, so it leaves some chemical cues that cause the leaf cells, not the insect cells, but the leaf cells to build these galls. So look at this giant kind of orange and red spiky thing or something like this, these incredible, beautiful constructions. We would have absolutely no idea that these flat green cells that reliably build this kind of leaf structure are even capable of this. Okay, what else are they capable of? So it's really important to start to understand that the default that we see every day isn't the limitations of the material, and that lots more are possible if you prompt the system in particular ways. Here's something else that's interesting and that's possible with this material. Here's a tadpole. So this is a larva of a frog, and this tadpole, there's the mouth, the nostrils, the brain, the gut, and the tail out here. What we've done here is we've placed some cells that are going to become eye onto the tail of these embryos. They go ahead and they build a perfect eye, and these eyes make an optic nerve. The optic nerve does not connect to the brain. It comes up, sometimes it synapses onto the spinal cord, sometimes on the gut, sometimes nowhere at all. But one thing you can find out from this, if you prevent the primary eyes from forming, is that these animals can see perfectly well. We've built a system that trains them on visual cues, so you can actually prove that they can not only see light, but they can learn to see and exert certain behaviors when they see certain things. And so this is quite remarkable because this animal has a completely radically different sensory motor architecture. The eye is now not in the head where it has been for millions of years. It's on the tail. It's sending information to who knows where, certainly not directly to the brain. And yet the animal can see. Why does this not take many rounds of evolutionary mutation, adaptation, selection of the best ones? Why does this work out of the box immediately? You make the change, everything works. And we could talk for hours about this, but the simple answer is that even normal embryos, even normal default embryos, are doing what this thing is doing, which is solving problems directly out of the box. Evolution does not make specific fixed solutions to specific environments. What it makes is problem-solving agents, and they solve problems in a number of spaces that we'll talk about. And both normal embryos and these kind of altered constructions have to figure out from scratch what they are, how to get along, and what they can do with the materials that they've been given. And that's because both on the evolutionary scale, on the behavioral scale, and on the cognitive scale, there is a constant process of creative reconstruction. And what I mean by that is that let's take the cognitive side first. The memory, any given creature at a given moment in time does not have access to the past. What you have access to are the engrams, the memory traces that the past has left in your brain or body. And so what you need to do at any given moment, and we are continuously, as living beings, we are continuously doing this, we are reinterpreting the molecular evidence that is in our bodies, the memories, and trying to actively construct a story of who we are, what we're doing, and what our next move is going to be. This is essentially a constructive story that is also true in, for example, in evolution. So every embryo inherits inherits very compressed, narrow experiences that the past generations have had. They get this generative seed in terms of the genetics and the other components of the maternal cytoplasm that they get. And then from this, they have to reconstruct how they're going to live life. And so they take that information seriously, but they don't take it literally, as we can see from many examples of incredible plasticity, and I'll show you some later on. So this fundamental idea of continuously reconstructing what you are and what you can do from the evidence that you have is a creative process, because in this bottleneck, and this is a very familiar architecture to machine learning folks these days, it's a kind of autoencoder where you squeeze the data down into a very narrow representation in the middle. That happens because you don't store all your previous experiences. What you store is a generalization, right? You learn, that's the whole point of learning, whether evolutionarily or behaviorally. You learn and you compress everything that you've done into generative rules. You throw away a lot of the detail, and because that detail is missing, this process, the encoding process, can be deductive and algorithmic, but the decoding process has to be creative because you're missing a lot of that information, and you have to take that rule and say, but how does it apply now, and what am I going to do next? And all of that can be seen. So this remarkable ability to deal with uncertainty and having to put together a new life in the world every single time from scratch is something that we all go through. We all start life as a single cell oocyte, basically of a little quiescent blob of chemistry and physics, and then slowly through this incredibly remarkable process of embryonic development, we become something like this, or even something like this, right? And so what's really important is that we have to remember this as people try to make very firm distinctions. Well, humans do this, and chemical systems do something else. Well, we were all a chemical system once, and there is no special point during this process during which our mental capacities sort of snap into focus. But we need to develop our slow and gradual our models of slow and gradual scaling of the kind of capacities that these systems have into the kind of capacities that we have as functional adult humans. This is a gradual process of enlargement and projection into new problem spaces. And even that's not the end of the story, as I'll tell you about today, via cancer and via a weird kind of life after death that's possible in terms of novel biological constructs made from our body cells after development. So what this is reminding us of is that really what we owe are stories of scaling and transformation, not categorical differences between things, but scaling and transformation, because we are all made from an agential material. This is a single cell. Okay, and this is a free living organism known as a lacrimaria, but we are all made of single cells. And notice how incredibly competent it is at single cell agendas. It handles all of its physiological, metabolic, behavioral needs in one cell. There's no brain, there's no nervous system, there no stem cells, just one cell handling its business. And it's these sorts of things that have to work together to create whatever it is that we are. And so that's a little disturbing that we scale from a passive chemical medium into these kinds of active cells and then into whatever we are. But at least, many people think, but at least we're a true unified intelligence. We've got this nice solid brain. We all feel like a unified intelligence most of the time. And ants and bird flocks and bee colonies, which Ricard Soleil calls liquid brains, we tend to think those are just metaphors. Kind of these things are collective intelligence, but we're a real solid intelligence, right? Well, that's actually not true either. So, for example, Descartes really liked the pineal gland because there's only one of them in the brain, and he thought that was associated with the unitary integrated experience that humans have. But he didn't have access to good microscopy. If he had a good microscope, what he would have done is looked inside that pineal gland and said, wait a minute, there's not one of anything. The pineal gland is made of tons of cells. And if you look inside each one of those little cells, you find all of this. So all intelligence is collective intelligence. We are all made of parts, and everything that we're interested in emerges from a story of how these parts connect together to give rise to higher level organization that has properties that the parts don't have. So our bodies are made of a kind of multi-scale competency architecture where all along from the tiniest components all the way through our bodies and then our whole swarms, there is intelligence at each level. And when I say intelligence, what I'm focusing on here is problem solving competency in various spaces. There are many different kinds of problem spaces, including the familiar behavioral problem space when we study rats and things like this. But there are many other problem spaces in which biological systems live. They traverse these spaces and they solve problems. And we need to understand how all of these things relate to each other to give rise to the amazing capacities that we enjoy. Now, I will point out, although I don't have time to talk about it, that there's even intelligence below the cell level. A lot of people sort of start this question about individual cells and whether individual cells can have some problem-solving properties. Well, it turns out that even within single cells, something as simple as a gene regulatory network, which is a simple chemical a set of chemicals turning each other on and off, already, just that alone, already has 6 different kinds of memory, including Pavlovian conditioning. So very simple systems can already do things that are on the spectrum of cognitive kinds of properties. So memory already starts down at the chemical level, possibly below that. And we are now taking advantage of all of that in the lab to try to train chemical networks in cells for useful properties, for example, with drug conditioning and other examples of biomedicine. So intelligence is really baked into our substrate, and these kind of capacities is really there from the bottom up. And humans are really not very good at recognizing all of these. We do okay with medium-sized objects moving at medium speeds through three-dimensional space. We can kind of recognize that as intelligence. But it's really important to know that perception action loop can happen in all kinds of other spaces, which has implications for unconventional embodiments. We'll talk about that shortly. And if we had the ability to feel, for example, our blood chemistry, well, the way that we see the outside world and the way we sense it, I think we would have no trouble recognizing that, for example, our liver and our kidneys are navigating, are an intelligent symbiont that's navigating various problem spaces to keep us alive every day. So the space of possible gene expressions, the space of physiological states, and all of these other spaces. We're just not very good at noticing all of the life and striving that happens in these other spaces. What I want to talk about next is some very specific examples about one specific kind of space. So this is anatomical morphospace. Anatomical morphospace is simply the space of all possible large-scale shapes that a group of cells can build. And so how do we get there from a single cell to a specific complex anatomy? Okay, so here's where I'm going to now start talking about the bioelectric interface to the collective intelligence of cells. But first, I need to give you a few examples of what do I really mean when I say collective intelligence. Well, when I say intelligence, I am not talking about necessarily a second order. What does it feel like to be solving problems? I know what goals I have. That's a high level intelligence. I don't necessarily mean that. I mean how William James defined intelligence, which is the ability to get to the same goal by different means. So in particular, how to reach your goal within a certain space, even when circumstances change. Okay, so I want to show you a very simple example. So here's a cross section through a kidney tubule in a newt, and what you'll see is about 8 to 10 cells that work together to form this kind of structure. Well, one thing you can do with these salamanders is that you can force the early cells to have way more copies of the chromosomes than they normally do. So you can make polyploid newts that have 4N, 5N, 6N, and so on, a copy number. When you do that, the cells get bigger because the nuclei get bigger to accommodate more DNA, but you still get a normal newt. So that's amazing thing number one, is actually you can have way more copies of your genetic material, no problem. It still builds a perfectly normal organism. Then the second thing is that, well, the cells get bigger to accommodate it, but the newt stays the same size. So how does that happen? Well, it happens because now fewer but larger cells make the exact same structure. So the structure actually adapts to the changing cell size, again, immediately, not through more rounds of evolution, but immediately adapts and gives you the same exact structure using bigger parts. Now, the really amazing thing happens if you make the cells truly gigantic, then just one cell will wrap around itself and give you the exact same structure. So now notice what this means. First of all, this is a kind of top-down causation, because in the service of having this particular anatomical outcome, different molecular mechanisms are getting activated. In this case, cell-to-cell communication. In this case, cytoskeletal bending. So this is a case of the system adaptively using the parts that it has in novel ways to solve new problems. I mean, this is a great definition of intelligence, using the parts you have in new ways to solve a new problem you haven't seen before. The other thing that this reminds us of is the amazing problem-solving capacity. If you're a newt coming into the world, what can you count on? Well, not only can you not really count on your environment, because many things can change, but you can't even count on your own parts. You don't know how much genetic material you're going to have. In fact, through evolution, you're guaranteed that the material will change because it will be mutated, but you don't know how much genetic material you have. You don't know the size or number of your cells. You have to complete your journey in morphospace with the tools you have, even when things change. This is the fundamental thing about living beings, is that they are constantly solving problems. And another example of this kind of thing is something that we discovered a few years ago, which is that when tadpoles become frogs, they have to rearrange their face. So the eyes have to move, the jaws have to move. And originally it was thought that, well, if you just specify somehow the direction and the distance for the movement, then you can just hardwire it, because every tadpole looks the same and every frog looks the same. And so if you just tell each part which direction and how far to move, then you'll get a normal frog. So we decided to test this. And this is a really important part of this kind of framework. When you're dealing with unconventional intelligence, you can't just make assumptions, you have to do experiments. And in particular, you have to do perturbative experiments to see how smart the system really is. So what we did was we created what we call Picasso tadpoles, where everything is in the wrong place. The eyes off to the, the eyes on top of the head, the mouth is off to the side. It's just scrambled like a Mr. Potato Head doll. Everything is in the wrong order. And what we found is actually you get pretty normal frogs out of it, because all of these components will move in novel paths. In fact, sometimes they move a little too far and they have to come back. They move in novel paths to get to where they're going. So this is not a hardwired scheme. This is actually an error minimization scheme. What the genetics actually specifies is hardware that can deal with novelty, that remembers what the final pattern is, and can take novel actions to get us there when it's starting at an abnormal starting position. So all of these things show the very specific problem-solving capacity. So the reason I call all of these things intelligence is not because of rising complexity. It's not because of reliability in development. It's specifically this. It's the ability to solve new problems that you haven't seen before using the tools that you have. So this asks us to solve two very important problems. One is, how does it remember what the correct pattern is? Right? We have some idea of how goal-directed memories work in neuroscience, at least to some extent. But in this case, how do these cells know what the correct frog face looks like? And in general, how do the cells cooperate so that you get the same outcome at the end? In three-dimensional space, we kind of have an idea of how the cognitive glue works. The reason that we are an animal and not a pile of neurons is because the neurons interact with each other in electrical networks that allow this kind of amazing thing. When you train a rat to press a lever and get a delicious reward, well, the cells at the bottom of the feet interact with the lever. The cells in the gut get the delicious reward. No individual cell has both experiences. Okay, there is no single cell that had both experiences. So who owns the associative memory? Well, it's the rat. And the reason you have a rat and not just a pile of cells that couldn't solve this problem is because of this amazing architecture. So in the nervous system, we have the hardware looks like this. So you have these cells that are connected to each other with electrical synapses. You have little ion channel proteins on the cell surface that allow the cell to create an action potential and keep a particular electrical state, which may or may not get propagated to its neighbors. And the trying to read out the software looks like this. This is a zebrafish brain. And where all the electrical activity is being recorded, and you can see that all the information processing that's happening here is the idea is to be able to read that out and decode it so that we can look at this electrophysiology and we can know what the animal is thinking about, what the goals are, the preferences, the memories, and so on. Right? That's the project of neuroscience, is to be able to read this information. And there's been some progress in neural decoding and things like that. Well, it turns out, and this is what we've been working on for many years, it turns out that this system is not unique to brains or neurons. It's extremely evolutionarily ancient. It developed around the time of bacterial biofilms. And every cell in your body has these ion channels, and most cells have these electrical connections to their neighbors, so they form electrical networks. And so now we can ask the exact same kind of question. What does the rest of the body think about? We know what brains think about. Brains think about moving you through three-dimensional space. But what does the rest of the body think about from embryonic development? And we can, as it turns out, this is an early frog embryo where you can do exactly the same kind of decoding process. And we can ask the question and test this hypothesis. Is it possible to basically appropriate all the tools from neuroscience? This means both the bench tools, but also the conceptual tools for how we think about the electrical networks storing memories, implementing goals, and other mental processes. Could we take all those tools and apply them to an unconventional intelligence? Could we frame morphogenetic problem solving of the kind that I showed you a minute ago as behavior, as intelligent behavior in anatomical space, the way that neuroscientists use these tools to study behavior in three-dimensional space? So in order to do that, we had to develop the first molecular tools to read and write these electrical states in non-neural cells. So we use, for example, voltage-gated fluorescent reporters of electrical state. And so here you can see we can actually make videos now of these bioelectrical states. So here's a time lapse of an early frog embryo. Here are some explanted cells. The color indicates local resting potential. And so we can now track these electrical states. we do a lot of computer simulation, starting with the ion channels and pumps that create them, to tissue-wide kind of properties that enable pattern completion and regeneration and other memory properties of the tissue. So we try to understand how these electrical networks actually process anatomical information. I want to briefly show you two examples. This is something we call the electric face. This is a single frame out of this movie with a voltage die. And what you're seeing here is all the electrical states that are involved when the frog embryo is putting its face together. So here, this is showing you the layout of where all the organs are going to be. So here's where the mouth is going to be. Here's where the eye is going to be, the placodes. These are the electrical pattern memories that direct the gene expression that is required to form these various organs. This is a subtle scaffold. It's a pre-pattern. It's a subtle bioelectrically implemented informational pre-pattern that tells the chemistry what to do and ultimately the anatomy what to do. Now, this is an interesting kind of cognitive glue in that it binds individual cells towards large-scale goals, such as building large organs. But it doesn't stop there. In fact, it's also a kind of cognitive glue between individuals. So what you can see here is that if we poke this one embryo here, so you can see the response, but these others receive it very, very rapidly. And in fact, here, if you poke this one, these two embryos at a distance, within about 30 seconds, they can feel it. And so it also binds the interaction in groups. And in fact, we recently found that groups of embryos resist teratogenic perturbations much better than individuals. So in fact, there is a kind of a collective wisdom effect going on here, where the group is better at meeting its goals than the individuals. Okay, so we now have the ability to track all these different things, but of course that's not enough. Observation is not enough. To really understand what it's doing, you have to do perturbative experiments. And for that, we again appropriated some tools of neuroscience and used them in non-neural tissues. So this means we do not use any applied fields. There are no electrodes, no fields, no frequencies, no waves, no radiation, no magnets. What we do instead is control the interface that these cells are normally using to control each other. That is the ion channels and gap junctions that are there to control this electrical network. We use pharmacology, we can mutate the channels, we use optogenetics, all the tools that neuroscientists use to alter the information content of existing networks. Okay, so now it's time for me to show you what happens when you do that. Okay, so what can you do when you communicate with a collective instead of the individual cells or the molecular networks? Well, the first example, and I only have time to show you a couple of examples. The first example is this. In that electric face pattern that I showed you, there was a little spot that says this is where the animal's right eye goes. So what happens if you reintroduce that pattern, that bioelectrical pattern, somewhere else in the body? And you can do that by injecting specific ion channel RNA somewhere else, for example, in a bunch of cells that are going to give rise to the gut. Well, if you do that, what you will get is an ectopic eye. Wherever you put that spot, that's where you get your eye. And that eye has all the lens, retina, optic nerve, all the same kinds of things that it's supposed to have. So from this, you learn a number of things. First of all, you learn that the bioelectric pattern is actually instructive, meaning that you can use it to call up other organs in other places. It actually carries that kind of information. The second thing you see is that we really are communicating with the collective, not the individual cells nor the molecular networks. We don't tell it how to build an eye. In fact, we have no idea how to build an eye. So tens of thousands of genes will have to be up and down regulated. Different stem cells will have to do things, arranged in particular ways. We don't control any of that. What we do is we specify a very large scale signal that says build an eye here. That's it. The rest of it is on the system. And so this is of course, a hallmark of intelligent systems is that you can provide a simple prompt and they have the autonomy to take it from there and figure out all the rest of the things that have to happen in order for that to take place. So we communicate at a very high level with a very simple prompt. Also, we found that, if you look at your developmental biology textbook, it will tell you that only the cells out here, the anterior neurectarum, is competent to become eye. Well, that's true if you prompt it with a master eye, so-called master eye gene called PAC6. That's true. It doesn't work anywhere else back here, but the bioelectric prompt does. So when we're thinking about the competency of these cells, we have to be really careful and humble, because we're really taking an IQ test ourselves whenever we talk about the competency of any kind of system. And the competency limitation may be on us and not on the cells, as you can see in this scenario. If you know the better way to communicate with them, you get better outcomes. And of course, what we also found is that the cells actually self-scale to the task. So here's a lens sitting out in the flank of an embryo somewhere. If you inject only a few cells, so only these blue cells were injected, they actually recruit their neighbors to build the whole thing, because they know there's not enough of them. We didn't have to teach them that. We didn't have to control that, manage that. They do all of that themselves. So what we're doing here is, but we're exploiting this bioelectrical interface to gain access to the competencies of the material, the top-down modularity, the self-scaling, all of these beautiful things are available because we found this interface for communicating with them. And we have a regenerative medicine program where we try to learn to regenerate limbs. There's an incredible problem with limb and organ loss in terms of biomedical suffering worldwide. So frogs, unlike salamanders, but like humans, do not regenerate their limbs in adulthood. So if you cut the leg 45 days later, you get nothing. We developed a bioelectrical cocktail that induces within 45 days, you've already got some toes, you've got a toenail, eventually a pretty good leg that's both touch sensitive and motile. You can see here, if you touch it, it can feel it. And one interesting thing is that the treatment is 24 hours. Then you get a year and a half of leg growth. You don't touch it at all during that time. So again, this is not micromanaging the cells. This is not scaffolds, 3D printing, stem cells. It's not any of that. It's convincing the tissue in the first 24 hours that it should go to the leg growth path in anatomical space, not the scarring path. And this is where I have to do a disclosure, because Dave Kaplan and I are scientific co-founders of a company called Morphoceuticals, and we are trying to do this now in mammals and eventually, hopefully, in human patients, using these wearable bioreactors as a way to deliver bioelectrical information to the cells to get them to build complex things. The next example I want to show you has to do with these planaria. I told you that in these flatworms, when you cut them into pieces, each piece gives rise to a perfect little worm. So if you cut this middle fragment, you always get exactly one head, one tail. And we can ask the question, so how does it know how many heads it's supposed to have? And what I will show you is that there is if we look using these dyes that this dye technology that we've developed, you can see that there's a pattern in this fragment that says one head, one tail. And so what you can do is you can revise that pattern, and we can go in and say, no, actually two heads. Okay, and we do this by manipulating the electrical potential of these cells using ion channel drugs, and so they develop these two spots. And so if you do that, sure enough, it builds an animal with two heads. This is not Photoshop or AI art. These are real animals. But here's something very important. This bioelectrical map is not a map of this two-headed animal. This is a map of this perfectly normal one-headed animal in which we have altered the bioelectrical gradient. So the memory, the pattern memory here, the pattern to which the cells are going to build if it gets cut in the future, is actually a latent counterfactual memory. It doesn't match the anatomy. These cells have this two-headed anatomy, but that's not what's happening now. Right now we have one head. In fact, the molecular biology says, yep, anterior gene expression in the head, not in the tail. But if you cut this animal, then the memory becomes triggered, and they build what the memory says, which is this. So a single body, a perfectly anatomically normal body, can store one of two different representations of what we will do if we get injured in the future, right? So this is a very simple kind of counterfactual memory, and you can have different pattern memories within the same body, probably many more, but this is the one that we've nailed down the best. Now, why do I keep calling it a memory? Because if you take these two-headed animals and you keep cutting them in plain water, no more manipulations of any kind, they will continue to regrow as two heads forever. Now, notice that there's nothing wrong with their genetics. We haven't touched their genome. Their genomes are completely normal. This information, much like information during conventional learning, is stored in the stable states of the bioelectrical circuit, okay? And you can rewrite it. You can say from one head to two. We can now go from two head. We can rewrite it back to one head. And when you reset it, it keeps. That's the basic property of an electrical memory circuit. It keeps the information. And this does not require any changes to the hardware. So the question of where is head number determined is kind of tricky. You might want to say it's in the genes, because by default, they build a hardware system that has this pattern that says build one head. But that's just the default. That actually, the genes actually don't determine this, because you can build a two-headed animal. And this is invisible to any kind of genetic sequencing or anything like that, because you're not going to see any difference. That's not where the difference is. And here you can see the video of these two-headed animals hanging out. And not only can we make different numbers of heads, but we can make different shapes of heads. So we can ask this planarian with a nice triangular head to build heads of P. felina, flat, or S. mediterranea, round. And there's about between 100 and 150 million years of evolutionary distance normally between these animals and this guy. But this hardware is perfectly capable of visiting the other attractors in the anatomical space belonging to these other species. In fact, not only the shapes of the head, but actually the shapes of the brain and the distribution of stem cells become just like these other species. So what we can see again is that what we have in the control of the body is a navigation of anatomical space, and that navigation is possible because the cells are connected by electrical networks that store rewritable, readable, and rewritable memories of where it is that they're going. It's a navigation task. Okay. And so the next thing I want to briefly talk about is the notion of the scale of these memories. Okay, for any kind of goal-directed behavior, including the very simple kind of homeostatic things like maintaining a pH or a hunger level, or the kind of thing your thermostat does in your house, you always need to store the set point. You have to remember what it is that you're trying to implement so that you can reduce the error between the current state and the remembered state. So single cells and molecular networks can store very simple kinds of memories, such as scalar numbers, such as pH range and things like that. But when cells get together, they can store actually a much bigger kinds of memory. For example, we're building a limb. No individual cell knows how many fingers you're supposed to have or what a finger is, but the collective absolutely does. And what happens during a development and during evolution is an enlargement of what I call the cognitive light cone, the scale of the largest goal a system can pursue. Okay, what is the largest goal in space and time that a system can pursue? So what you see here is that little individual cells have little tiny cognitive light cones because all they care about is the current conditions within this radius, right? Everything else, the environment, that doesn't matter, but it's keeping its own physiological and metabolic parameters within scale. But during development and during evolution, these cognitive light cones scale so that the cells can take on these incredibly large construction projects, right? So here's a salamander limb. If the salamander loses its limb, it will rebuild, and then the cells stop. They stop because they've reached the target, right? They know exactly what it looks like, and they've reached it. Not the individual cells, the collective. And so it's the network that can store this giant pattern and pursue these grandiose construction projects. Individual cells don't do that. But this process of scaling has a failure mode, and that's cancer. So when cells individually disconnect from the electrical network, they can no longer remember the large-scale goal they were working on. They revert to their ancient unicellular goals, and those being reproduce as much as you want, to metabolize as best as you can, go wherever life is good, and that's what you see in cancer in terms of metastasis. Okay, so this process can scale up, but it can also scale down. So the size of your goals during an individual's lifetime can grow and shrink. And these cells, I'll just point out, are no more selfish than normal cells. They just have smaller selves, right? So the size of the self, the size of the goal that these things are building is very large, and then it can shrink down. And so that boundary between self and world can shrink. Now, that kind of weird way of thinking about it has direct therapeutic implications. It means that instead of trying to kill cancer cells with toxic chemotherapy, could we try to force a reconnection of them with the rest of the network? And so when you do that, so this is a human oncogene injected into a frog embryo, it normally makes a tumor. And even before it makes a tumor, you can already see the bioelectrical abnormality here. The cells have disconnected with their neighbors, so you can already detect this. So of course, this is a potential diagnostic tool. But then what we did was we injected an ion channel that forces a particular bioelectrical state to reconnect them with their neighbors. It doesn't fix the oncogene. It doesn't correct the genetic damage. It doesn't kill the cell. It just reconnects it to their neighbors. And when you do that, the oncoprotein is blazingly strongly expressed. It's labeled with red fluorescence. In fact, it's all over the place. You can see it. Well, this is the same animal. There's no tumor. And the reason there's no tumor is because even though the individual cells are damaged, they have broken hardware, the collective is still working on what it needs to work on, which is to make nice skin and muscle and everything else. So you can see here the benefits, the practical benefits of asking what is the boundary of the self? These very philosophical questions about how selves demarcate themselves from the outside world. That leads quite directly to practical interventions, and this is now we're applying it, of course, to human patient samples and hopefully someday to real patients. So I want to also point out we've talked about the problem solving of collective intelligences. We've talked about the size of the goals of these collective intelligence. We've talked about rewriting these goals in terms of anatomical patterns that we can change. And one thing before I get to this final part of the talk is just to talk about where do selves come from and what decides the number of selves. So if we look at an early embryo, so let's say a human embryo, this is an embryonic blastoderm, there's maybe 50,000 cells. It's this kind of like flat sheet with 50,000 cells. What are we counting when we say that's one embryo? What is there one of? Well, what there's one of is a kind of alignment. It's the fact that all of the cells are connected into a network that is pursuing together a very particular path through anatomical space. It's going to make this nice embryo. So that's what we're counting actually here. But what you can do, and I used to do this in duck embryos as a PhD student, if you take a little needle and you just put some scratches in this blastoderm it takes about 4 or 6 hours for them to heal up. But before they do, each of these little islands doesn't feel the presence of the other, so it self-organizes into an embryo. And then when they do heal, you get conjoined twins. So you can get twins and triplets and so on. So the question of how many individuals are present within an embryo is not obvious at all. It could be anywhere from 0 to half a dozen. It's not set by the genetics. It's a self-organizing system in which multiple selves can form. And in fact, in human cases, there have been many cases, both conjoined and of course the typical non-conjoined twins, where multiple individuals exist in the same body. So one thing we have to ask in biology, which we know in computers how this works, we know how much processing can be done per number of transistors in your RAM, but in biology, it's a much tougher question. How many individuals per cubic millimeter of cells? And in fact, if I showed you a brain and you didn't know what a human was yet, you couldn't answer the question, well, how many human personalities could fit in here? Like we actually don't know what the carrying capacity of the medium is. And this kind of thing, this ability for multiple selves to organize in the same medium, and by the way, dissociate as well, has lots of implications for issues in cognition. So split brain patients, dissociative identity disorders, when you do have multiple personalities of different types within the same body. And for this reason, we actually study cancer as a dissociative identity disorder of the morphogenetic intelligence. It isn't about the broken hardware. It's really about the inappropriately shrinking boundary between self and world that creates a dissociation. Okay, and so what I showed you at the very beginning, so this is the last part of the talk. What I showed you at the beginning was this kind of spectrum where we somehow very slowly and gradually evolved and develop the kind of properties that we attribute to adult human cognition. But there's actually another spectrum, which is the ability to modify this system in novel ways, both technologically and biologically. Because the biology is so interoperable, and I showed you this at the beginning, where you can do all kinds of crazy things with these living systems, and they figure out some way of putting together a coherent being. You can actually introduce engineered materials at every level of living architecture. So within the cell, within the tissue, within the organism, and so on. You can mix chimeras between naturally evolved systems and engineered systems. They work together perfectly well. So that raises some interesting questions. If this kind of anatomical morphospace and behavioral space with specific attractors, we know where that comes from. It comes from genetics and environment. It comes from a long history of selection. That's what shapes these anatomical goal states. But what happens with these novel organisms that have never been here before, right? And how are we going to relate to these other beings when they are nowhere on the tree of life with us? Are we going to try to do this thing and try to figure out, well, is it 51% human or what? I mean, that isn't going to work because again, this is a very slow and continuous kind of axis. That doesn't make any sense. We need to figure out what are the properties of knowing novel beings, and where do they come from? Where do the patterns of shape and behavior come from? we typically think of genetics and environment, but I think we're going to have to go beyond this. And one way to start thinking about that is this kind of thing. What you're seeing here is a particular kind of fractal. It's called a Halley plot, and if you want to read more about it, you can read it here. That is revealed when you plot the properties of this very simple equation. So here, 6 or 7 characters, that's all it is. Inside of this little tiny formula is hidden all of this amazing structure. And if you vary the formula just slightly, you can make these videos of these kinds of things. All of this incredible complexity is sitting inside of this tiny little formula. It also doesn't hurt that it looks quite biological, right? This is kind of familiar kinds of shapes, but the whole point is that all of this is hidden in here. Now comes the interesting question. Well, where does it come from? You know, how do you explain this? How do you say where this pattern actually comes from? It's not, you know, you might give the formula, but is that a good explanation of where do these patterns come from? There is no genetics here. There is no environment here. There is no selection. What you're seeing here is that not just unpredictability and noise, but actually quite coherent patterns with specific subtly broken left-right asymmetries and so on arise from very simple prompts. And there is no feature of the physical universe that you can blame this on. This is not derived from any of the constants of physics or anything else that you, and there's nothing you can do in the physical world to manipulate it. It is simply something else. So now we have to ask ourselves, what about living things? Is this relevant to living things? Can we find living forms with no evolutionary history? What happens? Could you have patterns that do not arise from a pre-existing history of selection? So I'm going to show you a couple of examples of that. So if you look at this, you might ask, what is this? And looking at it, you might think, well, this looks like a primitive organism, like you got this from a bottom.
Video on it. Side Hello.
Excuse me, what? Somebody say something.
Sorry, that was a mistake, Dr. Levin. We've muted them.
Okay. You could think that this is some sort of primitive organism that you got from the bottom of a pond somewhere, and you might also take a guess at what the genome might look like, and so it might look like the genome of some primitive organism. Well, I can tell you that the genome here is 100% Homo sapiens. Okay, this is a little organism. We call them anthrobots. They're made from adult human tracheal epithelial cells. This is not embryonic. This doesn't look anything like any stage of human embryonic development. They're made from adult human patients, some of whom are deceased, some of whom are still alive, that donate tracheal epithelial cells. And we have a very simple protocol in which they self-assemble and they reboot their multicellularity. They turn into this kind of self-motile creature. So you would not have been able to guess the genome from this, and conversely from the human genome, you would not have been able to guess that these things are capable of doing that. One of the things they can do, which is quite interesting, if
Dr.
you play
Levin?
Yes?
Also, if you can just conclude, so we have a little time for Q&A. You've presented so much, and I know myself I have some questions. I'm sure other people might as well.
Okay, sure. Yeah, I'll finish up real quick. Yeah, it has the ability to heal human neuron wounds. I mean, who would have known that your tracheal cells, which sit there quietly in your airway, have the ability to form self-motile organisms that will heal neural wounds? Who knows what else they can do? I won't talk about their changes of their transcriptome and behavior. We'll skip some of this the Xenobot stuff. Here, and just to point out, okay, here's my final point, is that where do these behaviors and shapes come from if it's not evolutionary selection? There's never been any anthrobots, there's never been any Xenobots. Where do these things come from? Well, one thing people like to say is that these are emergent, and when you push on that and say, well, what does that mean? And say, well, they're just facts that hold about the world. And one thing you could do is simply record them when you come across them and be surprised each time, and just, okay, there's just a grab bag of things that hold. But another thing you could do is you could say, no, actually, maybe there's a latent space of form and function that can be systematically studied in a research agenda using these kinds of tools that I showed you today to actually map out that latent space of forms. It's not a physical space, as I showed you with that fractal. It's not determined by anything in the physical world, but we can have a research agenda of that space the way that mathematicians do when they study the space of mathematics. They don't think it's a random space of truths. They actually think they're studying they're exploring a pre-existing space. So one of the things that I've been developing is a way of using this idea and trying to map out that option space and that some of the things in that space are not simply geometric truths and the number theory and things like that, as has been said for many years, but actually specific kinds of minds, and that we can use this to these synthetic morphology as a vehicle for exploring that latent space. But one of the issues with this is that oftentimes, especially the organicist community, really thinks that, okay, that might apply to quote unquote living things, but that's it, right? The inanimate matter, it doesn't do this. And again, I won't have time to talk about this, but just to point out that first of all, that whole distinction is out the window because of all these potential hybrid forms, right? That exist, the space of possible bodies and minds is cyborgs and hybrids and all these things that we are going to be living with, and we need to develop better ethical frameworks for them, and some extremely basal forms that we have studied, for example, simple sorting algorithms that also do things, including problem solve in interesting ways that are not in the algorithm. So my claim is going to be that it doesn't take cells or life or huge complexity to have not just emergent unpredictability, but actually emergent cognition. And even very simple things do this. And so we need to be fearless in understanding that for the same reason that biochemistry doesn't tell the story of the human mind, the physics and algorithms of quote unquote machines don't tell the story of artificial cognition either. And then we have to remember that our formal models, such as Turing machines, have limitations, but they're just formal models. They don't actually cover what the system is capable of. And so I think, you know, the updated version of the Garden of Eden slide is going to look like this. We have a much wider world of very unconventional intelligences that we need to ethically relate to. And so I'm just going to end here, I guess, by thanking the people who did all the work. So these are all the amazing students and postdocs who did all this work and our collaborators. And then again, the companies that have supported some of this work. So I'll just stop here. Thank you.
No sound is coming.
Thank you very much, Dr. Levin, for your very in-depth and wonderful presentation. I'm sure that there are questions from the audience. We have about 15 minutes for that. If anyone would like to start. Otherwise, I'll start if no one else has a question.
Yes, Dr. Levin, you have wonderfully present your talk on self-constructing bodies, collective minds. And it includes some points from computer science and also in cognitive biology. And also you have presented the practical applications. And I want to ask a question, and this is my just a humble inquiry. You told in your presentation that life enables information to move across media. And you also told that newly regenerating brain can get information from rest of the body, right? But my question is, if body is not alive, then is it possible that newly regenerating brain can get information from the same body? Yes, please explain.
Yeah, you're asking whether a brain can get information from a body that is not alive. Did I get the question correctly?
Yes, yes.
Okay. Well, here's one thing we know. So, for example, David Glanzman's work at UCLA, he takes a sea slug, he trains the sea slug on a particular task. He then extracts the cells, grinds them up, extracts the RNA. So the original cells are not alive, the body is not alive. He takes that RNA, he injects it into a naive, untrained animal, and that animal then inherits the information, and it's able to show recall in behavioral assays. So from there, it's pretty clear that information can be encoded and transmitted in molecules. Not an entire living body. I don't know if you could do it with a body that's been quote unquote dead for some time. I have no idea about that, but it's clear that information can be moved in even simple chemical media like RNA.
Dr. Samangala, you're muted.
It's not actually, I think it's not a right, yeah. It is not possible that if a body can get information or RNA and DNA and get information from a dead organism inside the body. But living body can act how it can act. It is from our Vedic, Bhagavad Vedic paradigm, that that living body can, from the living body, the brain can regenerate the newly all information to create some other, or develop some other organism, or some develop inside the organism or the body, uh, only because of the, if there is a presence of soul. And the living body has the soul. It is given in the Srimad Bhagavad Gita, and according to the Vedic science, in Srimad Bhagavad Gita, it is given that the main source of all activity and function of the living body is the soul. And this is my humble submission. Thank you.
Yes, I'll pick. Oh, Dr. Roslenbridge, please go ahead.
Yeah, thank you for this very interesting talk. I really enjoyed that very much, and I'm very sympathetic to the idea that intelligence is really inherent on all levels, that is really a key. And it's not just intelligence within the DNA, but within the whole cell, and basically on all levels of the tissues. My question is, isn't it the case that there is always a sort of intelligence of the whole animal, of the whole system? So there is a sort of integrative system that is basically the owner of the intelligence. And many of your experiments, don't they show that things happen within this informed overall system? So it's a strong, or at least some examples are very strong hints at the coherence of a system. And that's a quite different picture than seeing the activity of a DNA that does something. So what I've tried to read in your experiments is this intelligence of the whole context of an integrative systems. Do you see this different, or is it about what you are looking for?
Well, so one of the things that we study are mechanisms of what I call cognitive glue. So these are policies that allow active intelligence subunits to also merge into a new collective that will have different goals, memories, and competencies projected into new spaces that the individual pieces don't have. So I think that's all true, but I think it's all simultaneous. So your cells are simultaneously solving little tiny local chemical problems, but they are also working together to solve the anatomical problem of maintaining your body. And the body is working on huge problems in three-dimensional space and maybe the linguistic space, and you and I might have cognitive light cones that project long after we're dead, because we're thinking about what the earth will look like 100 years from now, or what humanity will do a million years from now, whereas the other parts have smaller cognitive light cones that deal with more immediate environments, maybe smaller regions of prediction and memory and so on. All of that is happening simultaneously. And I also don't think that there's any guarantee that a collective system is more. I mean, it's difficult to say more because these are in different spaces, but the collective is not necessarily more intelligent than the parts. In biology, that tends to be true, and biology is very good at scaling up the intelligence of parts towards a larger mind of the collective, but it's not guaranteed, and it's pretty easy to make collectives out of intelligent parts that are actually not very bright at all. So the question is, how do these cognitive glues work? And we have quite a bit of detail about it now to give rise to these emergent beings.
Yeah, that's important to say that it is interdependent. It's on different levels, and every level has its intelligence, and that makes the whole system, all the parts, very flexible. And these parts are much more flexible than has been thought in the area of looking only at the DNA. And this flexibility is basically the basic stuff of evolution.
Exactly, yes. And we've done quite a bit of work showing how evolution works a lot differently when you're dealing with an agential material.
Yeah,
so
right.
So evolving over a hardwired material is not at all the same as evolving over an agential material. And that's correct.
That's extremely important, I think. That's really
Yeah.
a new view in biology. So thank you very much for this.
Thank you for that. Thank you.
Dr. Levin, I just want to say first, I think your work is very inspiring, and I look forward to the medical applications of it, especially. I think, you say very clearly that you're interested in philosophy to the extent that it helps the actual application and development of the science, and your work clearly has applications in regenerative biology and other medical fields, so we hope that can help many people. So my question is going to pick up from where Sumangala was going, I believe, Dr. Sumangala, where that the RNA in your example it can hold information from a dead body, but in order for the information to be decoded, so to speak, it needs to be within a living agent. So my question is about this denial of categorical differences that you bring up. There still seems to be a categorical difference between the living and non-living, because the examples that you give of a gene regulatory network having memory and cognition is occurring within a living entity, a cell, or even from the developmental point of view, when a living entity is just a blob of chemicals, so to speak, it's not just a blob of chemicals because it's coming from a living entity and it endowed with the potential for the development. So although it's an unmanifest potential at the stage of a blob of chemicals, it's much different than inert chemicals outside of a living entity, isn't it? So there still seems to be a categorical difference between living and non-living, where the living is characterized by being for self. It's always utilizing, whether it's inert structures or other living agents, it's utilizing it for its own means. And non-living structures don't seem to have that capacity. They have no being for self. So what do you think about this?
Okay, so let's analyze this. The first thing that I will claim is that all of these kinds of categories are only useful and should be maintained to the extent that they help make new discoveries. And so I'm currently actually working on a paper with I canvassed about 50 or 60 leading scientists in the space to give a definition of what distinguishes living from not living, and try to point out why that definition is useful for anything. And so the first claim that I'll make is that I've actually, and it's maybe weird to say for a biologist, which I am, but I've not seen those kind of definitions do any useful work whatsoever. In other words, but trying to make a crisp definition between non-living and non-living, I've not seen that move anything forward. If you, on the other hand, if you start to look at transitional cases, there are lots of cases that I think would actually be very hard to say something about. For example, in cryogenics, in examples where we have dissociated these kinds of systems into the components. What you said before about gene regulatory networks, actually no, they don't need to be inside of cells. You can do it with a simple 5 very simple chemicals sitting in a petri dish of water. They don't even need to be biological chemicals at all. In fact, you can do the same thing as other colleagues have shown with systems of connected springs, metal springs. So those kind of dynamics, those kind of learning dynamics, do not require anything that we would traditionally call alive. And I've seen a lot of people argue a lot about what these transitional cases, about what is alive and what isn't. What I haven't seen is that distinction move any research forward. So I think we should be maintaining categories that are doing some useful work and helping us to make predictions. I've not seen that for life, for life and not life, and giving a definition of what you mean by life is really hard. Like I said, if you email me later, I'll send you a link to the paper once it appears. All of these definitions, the 50 or 60 folks who work in this field do not actually agree on what the definitions are, because it's actually really hard. It's really hard to give a useful definition. So I would maintain that you think about what work you want that distinction to do. What is it doing for us?
Okay, Um,
Can I say question? Because
sure.
otherwise,
Or please not. Please.
so, thank you, Dr. Levin, for your nice talk. I want to ask you, when you say there is not necessary of making any distinction between life and death, then you are saying that a person is murdered, then that is as good as living, because why you call him dead? So there is some difference exists, it seems, living and dead in our normal day-to-day observation also. And the second thing is that machine, we never seen a machine which has its own self-identity and its own goal-driven things. A machine is always run by a person who designed it or programmed it or the owner. Machine don't have its own self-identity base running. A smartphone we call looks only smart when the person operating is smart. So we do see there is, the laws of nature which drives the inert matter, and the animated inner stuff, it violates that law. So there is clear distinction that is observable.
So I would push back on all of that. First of all, trauma surgeons over the years, their goal is to shift the boundary. Things that used to be considered dead are no longer dead. If you end up in the emergency room, if you did that 100 years ago, they would have said dead. Now there's reanimation further and further, and so their job is specifically to erase that distinction. And when you talk about machines, the cell phone is not the machine I'm talking about. First of all, as I mentioned in my talk, we have a very rich variety of hybrids coming. So when you see a human that has all kinds of altered components, are you going to ask how are you going to say that this is a machine versus a human? Are you going to measure to say, well, it's got 51% electronics in his brain, so therefore it's a machine? It isn't that simple. What you're thinking of very kind of classical ancient machines where it was easy to say, oh yeah, this thing's a machine. It's boring. It doesn't have its own its own goal. It doesn't have the capacity to set its own goals and so on. It used to be easy. We have to dump this going into the future because we are at a risk of massive ethical lapses by considering and treating as machines beings that are unconventional. They are hard for us to recognize, and they have moral worth. I think it is extremely dangerous to label these categories when the science and the technology are going in a place. I mean, we already have a long history as humans of treating other humans as if they're not full humans. They don't have they don't have real pain perception. Certain kinds of people, including women, get less analgesia. Right, this has been studied again and again in hospitals because people thought they don't feel pain the same way that men do. This is a very dangerous ground ethically, and I think we have to go beyond simple categories of life and machine, which were okay, in the 1900s. That was fine. This is not going to do for us, and it's for two reasons. One is because of the existence of hybrids and cyborgs, where the technology and the human are going to be joined together, and it is not going to be possible to say what of these ancient categories they belong to. And also because look, there's no reason why evolution has a monopoly of making real minds that matter in an ethical sense. If you think about what evolution is, for years it's been randomly poking around the biological material, and that's great, and here we are. But why is that the only way to make real minds? Why can't you create, not the cell phone, but in the future, with technologies that we already have now, why can't you, including evolutionary algorithms, why does it have to be the random pokings of cosmic rays to design real minds? No, engineers can contribute to this too, and we have to be open-minded about this. Otherwise, we are going to risk very serious ethical lapses. And a final thing, I don't talk about it because I don't have any experimental way of addressing it, so there's kind of no point. But I'll just mention on the theory that Dr. Sumangala was talking about the soul and things like that. If you believe in those things, and you believe that those things are in some way connected to the physical body that we are building, who are we to say that those kinds of things cannot find a home in new hybrid or fully artificial constructs that we make? If you really believe that there are souls that connect to physical bodies, I mean, evolution's great and everything, but why can't we make them a nice home and have them embodied by some other kind of spirit? I don't see any reason to rule any of that out.
I myself an engineer and I use thermocouples to measure the temperature. I never find a thermocouple feeling the sense of heat. It only gives the reading, but never experience any heat
You
or
have
a
no idea what it
you know,
experiences.
thermometer. So the
You have no
experience,
idea what it experiences.
you have idea because you experience it.
Well, you don't know what I experience either, and you don't know what the thermocouple experiences. And if you think that the thermocouple doesn't experience anything, then you have to tell me where from the journey from a single oocyte, which is just a bunch of chemicals like your thermocouple, to a human, you have to tell me where this sense of experience comes in.
That is the thing that scientists have to tell. They are claiming that molecules give a rise to experience. We are not claiming that. We are saying that experience comes from a very different category that is termed as soul. The empirical symptom of existence of the soul is consciousness. Consciousness is not the property of anything that physics and chemistry talking all these days,
Oh,
not
I
the
agree with
biology.
you. So on that one, I agree with you. But I think if you believe that it's a property of the soul, then you have to tell me which kinds of physical embodiments souls do and do not inhabit. And I don't see any reason to say that they're going to only inhabit things that are the result of biological evolution. And if we make a beautiful new body, that no soul will want to inhabit it. I don't see why you could possibly know that.
The science of soul is a very detailed topic, and maybe because of time constraint, we cannot discuss that, but is another topic of discussion. But as far as your explanation that machine has some experience of sense of heat or cold or happiness, no machine has ever has done that or anybody claimed that.
That's that's great, and that's exactly what the modern world says about the human, right? If you notice that's precisely the modern scientists say that the human is a biochemical machine, and therefore all these things that you're talking about are convenient fictions. The way that we come up with things that make it easier to have society and so on, and for the exact same reason that you don't buy that story for biochemicals, I don't buy that story for any kinds of physical bodies at all, for the exact same reason.
Perhaps this, any other discussion perhaps can happen at the end of the conference during the interdisciplinary dialogue. These are all very important discussions to have, and we're grateful to both Dr. Levin and all the participants for coming together to speak about these things in a respectful way. But if we can move forward to the next talk, Dr. Halkadale is patiently waiting. Thank you again, though, to everybody.
Thank you.